In this lecture, we're going to talk about Parkinson's disease, specifically pathophysiology and non-human models for the disease. First of all, we have to say explicitly that most cases of Parkinson's disease have an unexplained cause. In general, people suspect that dopaminergic neurons may be selectively vulnerable because the cell body of a dopaminergic neuron must maintain large amounts of axoplasm and presynaptic proteins. Here's the cell body, and as you know, it projects to many areas of the central nervous system, many parts of the brain. And so it is maintaining an enormous amount of cytosol in its axon and at its axon terminals. And this puts a tremendous burden on the cell body, which must then be maintained for many decades of life. Specifically, dopaminergic neurons may also be selectively vulnerable. Now, of course, there are many types of neurons that have large axons. And in fact, there are neurodegenerative diseases that correspond to most of them. The motor neuron in the spinal cord is susceptible to amyotrophic lateral sclerosis, which we will not have a chance to discuss in this course. The large Purkinje cell of the cerebellum degenerates in some ataxias. And as we've mentioned, the large cholinergic neurons of the basal forebrain tend to degenerate in Alzheimer's disease. Why each of these is selectively vulnerable in a certain disease is not understood. Dopaminergic neurons may be selectively vulnerable in some cases because dopamine is highly reactive. Here are these two adjacent hydroxyl groups, a catechol group, and they are selectively accessible to oxidizing agents, and they then form reactive oxygen species themselves. And so over a period of decades and decades, one has a buildup of reactive oxygen species inside neurons. Another cause of Parkinson's disease seems to be pesticides. There's a very nice paper by my friends at UCLA on pesticides in the Central Valley of California, and people who are selectively exposed to those pesticides who have a higher incidence of Parkinson's disease. And then there is the frozen addict episode. Several decades ago, in the San Francisco Bay Area, there was an impurity in synthetic heroin, which was taken up by the dopamine transporter. As you'll remember, the dopamine transporter is expressed only in dopaminergic cells. This impurity was changed to another compound, and it killed dopaminergic neurons quite drastically. Even before then, the influenza pandemic, worldwide epidemic of 1918, killed 20 million people. In many people, the flu specifically killed dopaminergic neurons. Some of the survivors of that epidemic lived through the 60s, and they were studied when Oliver Sacks was a young doctor in the Bronx, I believe. And that was the story of his book called Awakenings and of the film based on that story. He has gone on to write many wonderful books at the intersection of neuroscience and medicine. What about the proteins that seem to be defective in Parkinson's disease? Well, the best known is alpha-synuclein. It has an unknown function, although, as I told you, it seems to be involved in the trafficking and fusion of membranes, especially in organelles. An interesting aspect of alpha-synuclein is that it's an intrinsically disordered protein. That is, when studied on its own, it doesn't seem to have a well-described shape of alpha helices and beta sheets. It seems to be disordered. And mutant alpha-synuclein forms fibrils. So here is the monomer, natively unfolded. A bit of a tendency to cause a fibril, a long extended protein. And then we get an amyloid-like fibril. And then it is possible that these mutant synucleins can condense into Lewy bodies. The fact that a protein is intrinsically disordered does not mean that it's always disordered. Apparently, alpha-synuclein can become ordered, but only when it binds to certain other proteins. So it's the binding to other proteins which order synuclein. Interestingly enough, there are some data suggesting that alpha-synuclein can be transmitted among cells. The hypothesis has arisen that it can be transmitted like a prion, so that an improperly folded state in cell A could be transmitted to improper folding in cell B. We'll have to see how this field of research plays out. Another major theme in Parkinson's research is improper mitochondrial function. This large cell body, which must maintain massive amounts of exoplasm and also large dendrites, places severe metabolic burdens on the exoplasm, and the most metabolically active organelle is the mitochondrion. And so early mitochondrial function may be one of the causative agents in Parkinson's disease. Now, to study all of these problems, there are animal models for Parkinson's disease. Among the earliest animal model was Drosophila, fruit flies, which overexpress synuclein. And here is an example of 4 dopaminergic neurons, which when they are overexpressing synuclein, especially one of the synuclein mutants that are associated with human Parkinson's disease, these neurons begin to degenerate. While they're degenerating, they also show dense structures, which are like Lewy bodies. And indeed, the flies show a movement disorder. Here is a measure of the climbing ability, very much like the experiment that Seymour Benzer did with flies climbing up the side of a test tube. The control flies retain this ability until they're about a month old. But the flies which have been induced to express either wild-type synuclein or especially one of the human mutant synucleins lose this ability to climb at a much earlier age. So the flies have a movement disorder. There are more models for Parkinson's disease. Because of the toxin angle, there are toxin-treated mice and rats and monkeys, which have Parkinsonian-like tremors and movement disorders. There are a number of mice whose Park genes have been altered. We, in my laboratory, were working on one with alpha-synuclein, and these show varying degrees of movement disorders and Parkinson-like symptoms. Interestingly enough, it's been possible to show pathology in yeast that have been induced to express alpha-synuclein from humans. Yeast do not ordinarily express alpha-synuclein, but when they are forced to overexpress synuclein, and in particular mutant synuclein, they show defects in cellular stress that have also been associated with Parkinson's disease. And then, of course, there is the question of whether one can culture cells from people carrying Parkinson's disease mutations, disease in a dish, if you will, and begin to study some of the most basic processes out of contact with other cells that might have extraneous influences. Disease in a dish is a very powerful concept in modern science, and especially in neuroscience, because the brain is so complex. It's a great challenge to study the characteristics of individual cells in the brain. The 2 ways that scientists are approaching this use either human embryonic stem cells or human-induced pluripotent stem cells. Now, this is not a course in stem cells or regenerative biology, but the major challenge here would be to take cells from a person known to be at risk for Parkinson's disease and to perform experiments on these cells in a sustained and rigorous manner. And if, for instance, there are embryonic stem cells that have one of the mutations for Parkinson's disease, this would be an excellent preparation. A somewhat more foreseeable way to get these cells would be from skin cells from humans who have Parkinson's disease, with or without one of the defined mutations, to induce these skin cells, so-called fibroblasts, to go back to being stem cells, and then to induce these stem cells to become dopaminergic neurons. Now, there are still major technical issues in generating dopaminergic neurons that behave like vulnerable neurons, either from embryonic stem cells or from induced pluripotent stem cells. Pluripotent means able to differentiate into any other cell type. What is required are a number of growth factors in the cell culture medium, and these are maintained for at least a month. And neurons then begin to show some characteristics that suggest that they are dopaminergic. Of course, what you would really like to do is to distinguish between neurons that look as though they are substantia nigra pars compacta, the vulnerable ones, versus the ventral tegmental area, the non-vulnerable ones. Experiments of this sort are just beginning based on markers, genes that are expressed by one or the other of these dopaminergic populations. And then, of course, it's not enough simply to say that a cell has epitopes, molecules that can be addressed by antibodies for the classical enzymes such as tyrosine hydroxylase, because cells that make dopamine and cells that extend their axons to many regions of the brain have a very large number of specific characteristics that are not shared by other neurons. And of course, one asks which of those characteristics render them typically vulnerable to Parkinson's disease, and how does the vulnerability progress? So, disease in a dish is an exciting set of opportunities, but we need a lot more research. What about biomarkers for Parkinson's disease? Well, there is no effective blood test for Parkinson's. So an experienced neurologist is still the best judge of whether or not a patient has Parkinson's disease. An excellent criterion used by many neurologists is responsiveness to L-DOPA. If a person is able to move better, then that is a good indication that he has Parkinson's. Recently, an imaging reagent has been approved by the U.S. Food and Drug Administration. It's called ioflupane. It's a high-affinity dopamine transporter, DAT ligand, and it's been labeled with iodine-125, which is an excellent marker for single-photon emission computed tomography, or SPECT. So, in general, neurons that are dopaminergic and alive and healthy express lots of the dopamine transporter, as we have described in previous lectures, in order to take up the dopamine that's been released and to package it into vesicles again. Neurons which have died past the point that they can sprout new dopaminergic terminals have less of the dopamine transporter. And here are images using the iodinated probe from a normal striatum. These are SPECT images. This would be a lot less extensive in a patient with Parkinson's disease. So one of the major uses for ioflupane would be to differentiate symptoms that are Parkinsonian but may not be Parkinson's disease. You may remember in the video during our last lecture that the presumed neurologist was very careful to say that this was a Parkinsonian patient with symptoms as in Parkinson's disease. So you really do need post-mortem material to show that a person has Parkinson's disease. There are lots of behavioral symptoms that are called Parkinsonian that may not arise from deterioration of dopaminergic neurons. So one way to make that distinction is to use this iodinated ligand. And for instance, this might be able to differentiate Parkinson's disease from other symptoms such as essential tremor. And then we have to admit that ioflupane has not yet become effective as a screening or confirmatory test, or for monitoring disease progression, or for monitoring response to therapy. And this is one of the major uses that we could make for an imaging reagent. It would be to monitor either protection in the long run or symptomatic relief in the short run. And that, of course, will be the twin topics of our following lectures: symptomatic relief in the short run and protection in the long run.